Hello friends, myself Dr. Rakesh Kumar Tekade, and today I am going to talk about Dendrimers. The motivation for this lecture came from various conferences where I met several investigators asking me questions about Dendrimers, some basic uh, in nature, somewhere very inherent, somewhere very detailed, but very interesting. So with this lecture, I thought of sharing all those questions along with their answers. I'm sure this lecture is going to be very interesting, especially to those investigators who are willing to work in the field of Dendrimer. So ready, steady. So let us start with our slide. Just give me 1 minute, please. Thank you. So here are we now. Okay, so the topic of our today talk is Dendrimer as carrier, drug carrier, things to know. Before I start, let me help you to identify from where actually I am speaking and from where I started my career. So I basically started my career from Sagar University. From there, I got chance to go for Commonwealth Fellowship in United Kingdom. From there, all the way, I traveled via France. Then I went to US for postdoc. From there, Hawaii, Texas, and then ultimately I am here in this campus, in this beautiful campus of International Medical University in Kuala Lumpur in Malaysia. So this is where currently I am based. So let us start first with the typical structure of Dendrimer. I will not go in detail as I know from the program schedule that Dr. Abhay is going to speak a lot about Dendrimer in my preceding lecture. Um, so as you can see here that Dendrimers are polymeric molecules. They are unimolecular micellar in nature. The basic motivation for the development of Dendrimer came from the nature where Professor Tomalia developed it for the first time. The name of Dendrimer itself arrived from two distinct Greek words, dendri and meros. Dendri means tree and meros means like. So you can see from the schematic that Dendrimer do resembles or its branch do resembles like that of the tree or a claw of a lizard or some ice flakes. So uh, this is a very versatile carrier. So what are various fields of Dendrimer application? So there's Dendrimer application is not restricted to drug delivery or diagnosis. It is now have expanded to gene delivery, solubilization, targeting, and there are several other biomedical application of Dendrimers. A few are listed here. So Dendrimers are a very versatile carrier that can be applied for various applications. So what are the things that makes Dendrimer so versatile? What's the key point in that? So we can see in this schematic diagram of Dendrimer, we can see clearly that the location A, this is basically the point called interior crevices where drug loading can take place. Dendrimer have several modifiable amine groups. Let's take an example of a liposome or a nanoparticle where we know that we can load the drug and we can also modify the periphery of those nanocarrier. But the basic differences between those carrier and Dendrimer is that Dendrimers not only having high drug loading propensity, but they do have huge number of modifiable amines or different surface group that can be conjugated with ligands as well as some imaging moieties. So this in all makes them very versatile nanocarrier because they can be modified for application in different field, different purposes. So usually people wonders or they ask which type of bioactives can be loaded inside Dendrimer. So the answer is that almost all type of bioactives ranging from drug, siRNA, DNA, nucleotide, DNA, RNA, plasmid, lot many types of cargoes can be loaded inside Dendrimer because of its versatility. It fit into the loading of various nanocarriers. But the point to be noted is that the loading efficiency might differ from one bioactive to the other, and it's chiefly governed by the type of bioactives that we are trying to load inside the Dendrimer. uh, for case in hand, if we want to load a drug which is having higher water solubility, and another drug which has low water solubility, then a drug which has low water solubility will attain higher drug loading inside the Dendrimer. The reason is that the Dendrimer has hydrophobic zones, or the core of the Dendrimer is hydrophobic, and hence it allows the hydrophobic drugs to get loaded inside it in an higher amount. Usually this amount ranges between 15 to 85% in a very general way, if we want to speak, um, but it is way higher, as I have experienced, as compared to that of the other carrier. But again, coming to the point, it differs from bioactive to bioactives. So how does the next question, which is generally came into our mind, how does Dendrimer drug complex release curve look like? Means how it release the drug. So in order to understand this, let us recall the concept of minimum effective concentration and maximum safe concentration. We all know very well that that for a drug to be uh, pharmacologically active, they should be having concentration between minimum effective concentration and maximum safe concentration. Uh, uh, just maximum safe concentration. If we specifically talk about the conventional dosage form, then we can see clearly that in order to maintain the drug concentration for a prolonged period of time, we have to give several dosing as I can show from this pointer. We have to give several dosing. We are, on the other hand, if we are giving the drug with the help of in the form of drug dendrimer complex, or in the form of a drug conjugate, as can be seen from this blue as well as pink curve, we can see that that we can sustain the release of the drug for a prolonged period of time. The point to be noted is that the drug release profile vary according to the uh, way by which drug is loaded. For instance, if we have physically loaded the drug inside the dendrimer, then we can see that that the release pattern, although it follows a sustained release profile, but it eventually drops. While on the case of drug conjugated complex, or the drug conjugated dendrimer, the drug can be released for an extended period of time as compared to that of the physical complex. So usually it follows a sustained release pattern. Now, next question is commonly asked: what are various routes by which dendrimers can be given? Is it IV only, or is it IM, or is it what? Is it true that dendrimer can be given by IV only? It's not, because dendrimer can be given by various route, and this was recently reviewed by Professor Jain and his group, and from that I recommend reading this article to everyone. In that, our group have exhaustively compiled reports on administration of dendrimer via various routes. For example, intravenous, intramuscular, transdermal, ophthalmic, as well as by oral route. I wish to share that recently we have developed a very innovative uh, oral administration of albendazole using Dendrimer in its mucoadhesive form. The idea was solely developed by Professor Jain, in which we have tried to merge, or we have tried to amalgamate nanotechnology and conventional therapeutics. We have tried to uh, punch mucoadhesive albendazole complex inside tablet, and deliver for the treatment of GI infection. This is currently under review, and I hope you will see the final draft soon. Um, if you would like to know more on this topic, I encourage you to discuss this with Professor Jain. I'm sure he will be very happy to share its outcome with you. So the conclusion is that Dendrimer can be given not only by IV but also by intramuscular, transdermal, ophthalmic as well as oral route. Although IV route has been followed more exhaustively as compared to that of the other route, but it do have other possibilities of application. Uh, a lot many time it was observed that people try to understand why dendrimer increases tumor localization. I'm sorry, I am trying to take you to entirely different aspect with my eventual slide, but again I wish to recall that the motivation is that with this presentation I am trying to share the most common hows and whys and wheres and whys of dendrimer in various areas. So the next why is why dendrimer increases tumor localization. So the answer is very simple, that dendrimer, like other nanoparticles, they utilize as enhanced permeability and retention effect. As we all know that dendrimers are of nanometric size, they usually their complex usually fall in the range of 2 to 6 or less than 10 nanometer in size. So let us talk first about cancer. So we all know that cancer cells are um, very selfish in nature. They try to grab every single nutrient that come into the body uh, uh, to uh, to attain access to those nutrients. So for this they develop certain number of capillaries or blood vasculature around them. These are called angiogenesis. So during this process, they do develop lot many angiogenic blood vessels, but those vessels are highly abnormal in nature. They have uh, some uh, some nanopores or perforations as can be seen. So these nanopores basically cancer develops so that they can get more access to the nutrients. Looking onto other aspect, dendrimers are nanometric size. They can get access uh, to these nanopores and can reach the dendrimers mass and can reach the cancerous site. But this cellular uptake is very important, not only the localization. So the next question is, does dendrimer increases cellular uptake? From the earlier slide, we know that dendrimer increases tumor localization, but the tumor localization is not only the aspect which is important for delivery of an anti-cancer drug. The important step after localization is uptake. So the next question is, does dendrimer increases cellular uptake? The answer is yes, dendrimer do increases cellular uptake. So please have a look at this particular zone. In this, we can clearly see um, association of dendrimer complex, which is shown in green fluorescent, which are fluorescently labeled dendrimers. So we all know that cancer cells are negatively charged, while dendrimers are positively charged. So these two distinct phenomenon helps dendrimers to form an association with cancer cells and finally lead to its cellular uptake. It was usually observed that the rate of enhancement of the cellular uptake usually lies in the range of 2 to 50 fold. It depends from formulation to formulation, because it depends upon the charge based interaction. Usually the dendrimer complex forms nano architect which are having zeta potential greater than 20 millivolt, and the zeta potential of cancer cells are negative. So this positive negative interaction helps uh, the dendrimer complex to get taken by the cancer cells. Now the next point is, what is the mechanism by which dendrimer increases cellular porosity or uptake? The answer lies behind the toxicity of dendrimer. Here, the dendrimer utilizes its weakness and makes their strength while increasing the uptake inside the cancer cell. So we all know that, for example, PPI or Pamam dendrimer, they have amine groups on its periphery. They are positively charged. So these positively charged moieties, they are very toxic towards the cell membrane. So upon coming in contact with the cellular membrane, they form nano holes. So these nano holes eventually help dendrimer to get access inside the dendrimer. They increases the porosity of or the permeability of cellular membrane, and finally leads to its uptake inside the cell. So what is the effect of pH? uh, this point is very important to understand in order to understand the endosomal escape or other release pathway inside the cell, because here from this slide we understood how dendrimer complex enters inside the cell, but what happens after entering inside the cell? To understand that phenomena, it is very important to understand the effect of pH on the surface charge of dendrimer. So this slide shows that uh, dendrimer attains highly positive charge under acidic pH, while they get deprotonated under alkaline pH. Usually dendrimers are two-thirds protonated under physiological blood pH, but when they enter at 4 degree or 4 pH, or the acidic pH that exists inside the endosomes, they usually get fully protonated, while if they encounters any alkaline condition, then they get deprotonated. So this is the take home from this slide, that dendrimer attains complete protonation when they encounters acidic condition, while if they get encounters to alkaline condition, then they get deprotonated. Now let us build some more rationale on the basis of this. So these are some of the mechanism which we reported in chemical review recently with Professor Jain. So from this schematic, we can clearly see that there is a stable electrostatic interaction between the loaded drug and the dendrimer interior amine groups. But while it comes in contact with acidic pH, which usually exist in tumors environment, we can see that this uh, electrostatic interaction, they got distorted, followed by the release of the drug. This is one of the recent investigation in which we investigated the effect of pH on the release of melphalan, and we clearly observed that under acidic condition, rapid release of the drug was observed, while under physiological condition, a sustained release was observed. So the take-home message is that yes, dendrimers are pH-sensitive unimolecular micellar architecture that can help in attaining pH-sensitive drug release, which is an inherent property of the dendrimer that makes them so and so appealable, so versatile. So does dendrimer help in endosomal escape? This is the point which I really wanted to share, because endosomal escape is a very important phenomena which we can clearly see has been neglected in so many reports on targeted drug delivery system. Our sole goal is to target the drug inside the cell, uh, but in the meanwhile we forget to entertain a very important phenomena which is called endosome escape, because we know that most of the time we are delivering sensitive drugs like siRNAs, miRNAs, or some proteins, peptides. Our sole goal is to deliver them inside the cytosol or near the nucleus in a stable format. Our goal is not only to make them suitable for uh, uptaken by endosome, because if a formulation doesn't escape from endosome, then eventually it will end up in lysosome, which is having a very aggressive acidic pH, which will eventually lead to its degradation. So endosomal is a very important phenomenon that should be addressed in all case of targeted drug delivery. So the question is, does dendrimer helps in endosomal escape, and how? So the answer is yes, they do. So, but how? So as seen here, that drug DNA or a drug protein or a drug siRNA complex, when it comes in contact with with the cell. After attaining endosomal escape, it ends in endosome. But as we have discussed recently in earlier slide, that upon coming in contact with acidic pH, dendrimer swells, they protonate, and they generate positive charge, highly positive charge. This positive charge of dendrimer, they repel each other to get increase their size. This increase in size of Dendrimer eventually destabilize the endosome, finally leading to the release of payload in the cytosolic compartment, which is the overall goal, which is the overall objective. So we can see clearly that Dendrimer is a versatile carrier that helps in a very beautiful pattern in attaining the endosomal escape. While in case of other nanoparticle, or I don't want to take the name specifically, but let us call them other nanoparticles, we have to add some extragenous facilities like cell penetrating peptides or other sort of arrangements we have to make them endosomal escape capable, right? So dendrimers, I would like to project that they help in endosomal escape. This is our recent report from our laboratory with Professor Jain, where uh, we have tried to develop rifampicin formulation and tried to assess whether dendrimer formulation helps in attaining higher drug uptake inside the cell, and we can clearly observe several fold enhancement in the drug uptake of rifampicin as compared to that of the plain rifampicin, whose uptake was less than 2%, even after increasing the concentration up to 14 microgram. While under same circumstances, we observed several fold higher uptake of rifampicin following its delivery using monocylated dendrimer. So we can see that dendrimers helps in increasing the drug uptake. Now, very important aspect, which I almost been asked by several investigators: are dendrimers safe? The question is, they are safe, but everything comes with some cost. So this is the cost with dendrimer, that is its inherent cytotoxicity. So we can see in this schematic that they are a bit cytotoxic towards red blood cell, and this basically figures from palmam dendrimer treated RBCs. We can see that they alters its shape. So somehow exploration is going on to combat or fight this particular aspect of dendrimer and make them more suitable for drug delivery. The below schematic that shows the effect of dendrimer on various on these are B47 cancer cell, and we can see that it distorts the cell as well. So being a dendrimer scientist, this is the next challenge to control their or to manage their toxicity, so that their effective or their versatile drug delivery attributes can be employed successfully. So yes, the conclusion is that they are, although toxic or hemolytically active, but the good news is that we can also the arrangements are there by which we can manage this. uh, the next question comes in our mind: any reports are available on hemolytic toxicity? So there are many reports. Looking onto its curiosity, we have recently compiled this in international, and the article can be found in International Journal of Pharmaceutics, that has been compiled by Jan et al. In 2010 recently. So we can see that dendrimer are hemolytically toxic, but the point to note that this hemolytic activity of dendrimer increases with increase in their generation. um, we can see that uh, third generation dendrimer or fourth generation dendrimer, they are less hemolytically active as compared to that of the fifth generation Dendrimer. In our recent report in biomaterials, in nanomedicine, and in pharmaceutical research, we have done a excellent comparative study between 3, 4th, and 5th generation Dendrimer. So let us go to our next point. We will discuss that in later. So how I can manage the Dendrimer toxicity? So you can manage the Dendrimer toxicity by modifying its periphery with several bioactives such as polyethylene glycol is there, polyglycerols are there, chitosan, albumin. There are so many bioactive polymers. If you coat dendrimers with those bioactives, then its toxicity can be masked to a great extent. So there are several reports available. Here are some examples, and I encourage all investigators who are working in the field of Dendrimer to go through the toxicity attributes of Dendrimer and how they can be resolved, whether you can come up with other innovative aspects to control this or manage this toxicity rather than or apart from these reports. So you can see that you can drastically reduce the Dendrimer's hemolytic toxicity after coating it with bioactive or biocompatible polymers like lactose, mannose, galactose, or chitosan, albumin, polyethylene glycol. Right. So let us move. Specifically asking about PAMAM or PPI Dendrimer. PAMAM and PPI because these are among the most explored excuse me, these are among the most explored classes of Dendrimer. So the question is, are 5.0 generation Dendrimers necessary for their engagement as drug delivery carrier? This question came in our mind in 2009, where we decided to explore 4, 5 along with lower generation Dendrimer like 3.0G Dendrimer on one platform and come up with some proof and quantitative proofs that whether 5.0G Dendrimers are necessary to use as drug carrier. So we have I encourage you to go and read the complete article which has been published in Biomaterial by us recently in 2015 and another in Nanomedicine in 2014. um, uh, Please drop me an email at rakeshkumar@imu.edu .my. If you are willing to read this full article and don't have the full access, I will be very happy to share this article with you. So coming to the point, are 5.0G Dendrimers necessary? So the answer is no. 5.0G Dendrimer are not necessary. You can also go with 4.0G Dendrimer. Let me bring your account that if we take an example of PPI or palm arm Dendrimer, then almost 99.99% of research has been done on fifth generation Dendrimer, while the lower generations like 4.0G or 3.0G are, I must say, neglected. So in this investigation, we came up with several exciting outcomes. We observed that the loading capacity of 4.0G and 5.0G Dendrimer, if we compare them, then 5.0G Dendrimer definitely leads as compared to that of the 4.0G or 3.0G. But the release property are compromisable because the 4.0G Dendrimer are several fold less toxic, while they are showing the similar, almost similar attributes or drug delivery attributes as compared to that of the 5.0G Dendrimer. I have included this figure from our biomaterial paper, in which we can clearly see that this fourth generation based melphalan formulation, they are almost equipotent as compared to that of 5.0G Dendrimer based melphalan formulation. So 5.0G Dendrimers are not at all necessary, and I encourage investigators to also give attention to fourth generation Dendrimer also and see if it can serve your purpose, because maybe you can be able to make the formulation more biocompatible, less toxic with 4.0 G Dendrimer, and that can be very clinically acceptable, right? So another question is why Dendrimer increases solubility? Does all nanoparticle increases solubility? The answer is no. Not all the nanoparticles increases the solubility. There should be some mechanism. So what is the mechanism by which Dendrimer increases solubility? The answer lies in the structure of Dendrimer. Let us take an example of PPI Dendrimer. We can clearly see that Dendrimers can load drugs inside its interior cavities, which we call as loading zone, while they have free amine groups. This amine group, they form hydrogen bonding. In our recent investigation, we tried to simulate the dendrimer complex positioning in presence of water with the help of ChemDraw energy minimization software, and we can clearly see formation of hydrogen bonding between water molecules and dendrimer, and this is the mechanism by which dendrimer increases the water solubility of the complex. They sequesters the water insoluble drug inside their core, and forms the water and forms a hydrogen bonding with water and enhances its water solubility. So are any examples of drugs solubilized using dendrimer available? Yes, there are many. You can find in literature. This has been recently reviewed in biomacromolecules. So you can see that with the help of Dendrimer, solubility of several drugs which are having very poor water solubility, like adriamycin, anthracene, pyridine, mefloquinic acid, paclitaxel, naproxen, ibuprofen, ketoprofen, and lot many examples are there whose solubility has been enhanced with Dendrimer, that to several fold. So there are huge potential in Dendrimer. Apart from loading, it also enhances the water solubility. So what are PEGylated dendrimers? Because these are also a very important class of dendrimers. So PEGylated dendrimers are a class of dendrimer in which PEG chains, polyethylene glycol chains, are attached onto the surface of dendrimer periphery. They have been used for various purposes, starting from enhancing drug entrapment, solubilization, sustained drug releasing, targeted drug delivery, ranging from drug delivery to multifunctional drug design. So there are several advantages with PEGylated dendrimer. So apart from making the drug more water soluble, uh, what are the other aspect that follows PEGylation? So one major important aspect is enhancement in drug loading. From here we can clearly see that as compared to that of the plain PPI dendrimer, the loading efficiency or entrapment efficiency of PEGylated dendrimer enhances almost double as compared to that of the plain uh, dendrimer. So it increases, or it PEGylation leads higher entrapment, we can say in a conclusively manner. So does PEGylation affect in vitro drug release? Yes, it do. As we can see from this report, that PEGylation sustains the drug for prolonged period of time as compared to that of the unmodified drug. The reason behind this is that PEG chain, or the surface attached or surface anchored PEG chain, they offers additional obstruction for the release of the drug. Hence it sustains the drug release profile. So how drug useful in drug and gene delivery? So basically it is the slide that will combine all the above discussed phenomena. So we can clearly see that dendrimers which are positively charged can form complex with negatively charged dendrimer, negatively charged DNA or siRNA or protein or peptide. This dendrimer DNA or dendrimer siRNA or dendrimer gene complex, in a nutshell, are positively charged. They can get endocytosized inside the negatively charged cell very easily. as compared to that of the plain DNA. Of course, there is also a limitation with plain DNA or plain siRNA, which we cannot deliver directly because it will be degraded by RNAs or different type of degrading enzyme which are present in the blood. So there must be some mode. So Dendrimer offers one of the useful alternative for the delivery of genes or DNA, right? So can we use Dendrimer for multifunctional nanoparticle design? Yes, we can. Why? The answer lies in its versatile architect. As we have discussed in our earlier slide, that Dendrimers are having modifiable surface group in addition to hydrophobic core, where we can see drug can be loaded, and the modifiable surface group can be conjugated with targeting moieties such as folic acid. They can also be conjugated with drug if we are interested in conjugating it rather than loading it because of some reasons. Uh, we can also track it with the help of some tracking devices. So eventually we can develop multifunctional dendrimer or multifunctional formulation with the help of dendrimer because of the availability of several modifiable groups on its periphery. Comparing this aspect with other nanoparticles or other nanocarriers where surface modification is the main challenge compared to that of those carriers, dendrimers are very convenient to develop multifunctional nanocarrier. So conclusively we can see, yes, dendrimers can be used for the formulation of multifunctional nanocarriers. This is another class of dendrimer which I call lock-in dendrimer or dendritic box, which has been developed by Major long back. So can we lock the drug inside the dendrimer and prolong its drug release for an even more extended period of time? So yes, we can do this by modifying all the surface amine group or all the surface group of dendrimer with some locking groups like T-box. This will form a small, very compact architect we call as dendritic box. So in which drug can be loaded. It will not allow the drug to pass through it easily because of a very dense periphery. I must say that this is one of least explored or one of ignored class of dendrimer, and I believe it needs more investigation on this topic. So let's hope more reports will come on this topic in future, and we will be able to use this aspect for some attributes in biomedical field. So until now, rose Bengal dye or some drugs like methotrexate have been explored for dendrimer aspect or dendrimer dendritic box aspect. This is very interesting report which answers which ligand is the best for cancer targeting potential. Usually we have seen reports on folate targeted dendrimer or dextran targeted dendrimer, galactose targeted dendrimer. Specifically speaking, my laboratory, we have done this in separate lab bench, but on one platform this report was not available. So we've been successful in undergoing this experiment. You can see detailed aspect of this report in 2011 nanomedicine journal article. I encourage you to access full article, and as expressed, I will be very happy to provide the complete article. If you are interested and don't have the full access, please feel free to email me anytime. So question is, which ligand has the best cancer targeting potential? We have just selected three ligand because it was getting difficult for us to handle more than three ligands at that time because of time as well as other resource constraints. So we selected three most explored ligand like folate, dextran, and galactose, and we observed that folate was leading the fight. It ended up with most high number of apoptotic cell, most high number of dead cell, most cellular uptake as compared to that of the other targeting ligand into account. Now, we have tried to deduce its mechanism, and we anticipate that this is because of most favorable ligand receptor interaction which took place between folate and the receptor existing on the surface of cancer cell. I encourage you to read this full article, and I wish to express that I am very interested in seeing more such type of research in which investigator can perform, say, 15 or 20 targeting ligand on one platform. So that will be more encouraging and very interesting. So let's hope such type of investigation in future. Um, so what are liposomal locked in dendrimer? These are very important class of nanocarrier in which dendrimers were locked in inside the liposomes. We all know that liposomes represent one of most successful class of nanocarrier which have not only showed higher loading, high sustained release, but have also crossed FDA approval, clinical trials, and currently in the market with the name of Doxil or Lipodox for several formulations. So in this investigation, what investigators tried, they tried to load dendrimer inside the liposome. So what I believe the most important and interesting aspect of this research is that the core of liposomes are lipophilic. That means hydrophobic drug cannot be loaded inside the core of the liposome. In order to load hydrophobic drug, they should go uh, on the on the lipid layer, and eventually it leads to leaching. So in order to avoid that, this phenomenon came into existence, this amalgamation came into existence. In this, what investigators proposed that they proposed formation of a dendrimer drug complex which is hydrophilic in nature, and then loading this hydrophilic complex inside the hydrophilic core of liposome. So you can see the beauty of physics, the beauty of combination of physics, concept, fundament, and the physicochemical properties of dendrimer. They all authors tried to put in one place to develop this nanocarrier called liposomal locked in dendrimer. Motivated from this story, we recently tried to develop dendrimer stabilized nanoparticle, which we can see in pharmaceutical research. This we developed with the help of in collaboration with Professor Abhay Singh Chauhan, who was in University of Wisconsin. The outcome of this investigation was very exciting. We could clearly see the loading of dendrimer inside nanoparticle, forming layer by layer ways, forming layer by layer fashion nanoparticle. We explored this nanocarrier for the delivery of hydrophobic drug paclitaxel, and uh, so this was the motivation from liposomal locked in dendrimer, which is nothing but the formation of uh, combined nanocarrier using dendrimer and liposome. So are dendrimers comparable or having similar attributes to that of the linear polymer? So the answer is no. They are way differing with each other in their properties. So this is just an example. We can see that polymers, they are linear in nature, while dendrimers, they are globular. Polymers have low structural control, while dendrimers can be synthesized with high uh, structural control with size ranging between 2 to 6 or 2 to 8 nanometer. Polymers, they can or cannot have good water solubility, while dendrimers usually have high water solubility. Polymers, they have low loading capacity, while dendrimers, they have very high loading capacity. So you can see, if we compare dendrimer and linear polymer on one balance scale, we can clearly see that dendrimer leads the weight and is a better polymer as compared to that of the linear ones. So this is very important aspect which we try to clarify in our recent report. So the dilemma was Dendrimer generation nomenclature. Do it need harmonization? Are they differing from each other from lab to lab? Are they differing from each other from publication to publication? The answer is yes. We observed, we were in touch with the literature related to dendrimers in several years, and together with Professor Jain, we several time encountered uh, this non-harmonization. We also discussed this aspect with Professor Donald Tomalia, who is the father of Dendrimer. I also spoke with Professor Donald Tomalia during a conference in France, and from there we thought there should be something that should be brought forward, and it is related to Dendrimer generation nomenclature. Uh, we can clearly see two distinct type of dendrimer generation nomenclature. In one nomenclature, the first generation of dendrimer that comes into account is first generation dendrimer, while some lab starts it from zero generation dendrimer. We personally believe that the nomenclature which is start from zero generation nomenclature is correct and should be harmonized and followed. Of course, there are some reports which have already been published, but we can take this precaution in our ahead publications. I encourage all investigators to read this article which has been recently published in Drug Discovery Today. Um, um, it's top the title of this report was Dendrimer generation nomenclature and need to harmonize, and you will read. It's a very small article but very interesting. So dendrimer generation nomenclature do needs harmonization. So if you are working in the field of dendrimer, please, it's our group's whole sole humble request to you to please follow harmonized and correct generation nomenclature. Thank you. And next point is usually I was being asked this question, that are there any focus area of research that you recommend for dendrimer research, because a lot of research has been done. So the answer is that in comparison to that of the property and versatility of dendrimer, I say very little has been done, and huge research is need to be done. For a case in hand, to young investigator, I recommend to explore on formulation aspect for nasal, ophthalmic, and transdermal route, which although have been investigated, but it needs more and vigorous investigation. Multiple drug delivery can be developed with the help of dendrimer, so we can focus on this aspect. Dendrimer aspects in cosmetics is also have not been explored to satisfactory level, and I encourage youngsters to read article by Khopade et al. And Chauhan et al., in which they have shown higher transdermal uptake as well as some anti-inflammatory activity. So uh, you can come up with some therapeutic cosmetic nano device, or not, if then with cosmetic nano device for the delivery of various anti-aging stuffs, for the delivery of various beautification bioactives, for the delivery of very antioxidant, for the delivery of various skin protectives. You can explore this carrier. You can also explore Dendrimer for development of new type of Dendrimer. For example, we have tried to develop citric acid Dendrimer once in our laboratory, but unfortunately that research got stuck somewhere. But I encourage all of you not to stuck, but to give a kick start. If you are interested in doing something novel in the field of Dendrimer, then this is also a very challenging but very interesting area of research, uh, that is to develop novel and improved class of dendrimers, right? So that you will give rise to, or you will give birth to a baby that can go in different lab and share its physicochemical or share its properties, right? Use of Dendrimer in diagnostic is also having huge propensity and should be focused. So question comes in mind, are any Dendrimer product in clinical trial? The answer is yes. Viva Gel, Superfect, Alerts, Stratus are some of the formulation that are in clinical trial. We can see in this slide that several Dendrimer formulation from different leading companies like Durex, in which they have proposed to use dendrimers as an HIV protecting reagent. They are planning to, they already disclosed this fact that they are planning to coat this class of dendrimer onto condoms and sell it as a HIV protecting uh, 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 mode. So a lot many thing is going on. Lot many products are in pipeline with Dendrimer. So will Dendrimers and Dendrimer polymers play a key role in nanotechnologies towards development of a sustainable therapeutics? The answer is yes, as can be evidenced from increasing number of patents and publication. After its development since 1981, we can see thousands and thousands of publication and patents are there. At the end, I would like to thank you and the organizers for this opportunities to share this attributes, share these facts about Dendrimers. I hope all of you might have um, uh, I hope you have enjoyed this lecture. So to summarize, I would like to say that with this discussion, I had tried to cover some of the most aroused questions about dendrimers, some of the most aroused uh, phenomenon about dendrimers that comes in our mind, whether dendrimer do this or not, whether dendrimer can be used for this or not, how we can be how it can be done, how dendrimer can be modified for this particular aspect. So I hope you enjoyed this lecture, and I welcome all of you to or I welcome all of you to feel free to write to me if you want to have a complete print of any of our publication, or you are interested in knowing more about Dendrimer, feel free to write me at rakeshkumar@imu.edu.my. I will be more than happy to answer your query. Once again, thank you everyone. Have a very productive and very pleasant uh, time with this conference. Uh, do try to utilize this time to discuss and share your ideas. I thank Professor Jain and all of the organizers, the team, to give me this chance to speak on Dendrimer, and I thoroughly enjoyed this. Thank you very much, and Have a nice day. Thank you very much. Bye bye.